will get started. So a few things I have to say. First of all, the lab this week is going to be more fly time stuff, but not much. So I kind of expect that everyone will manage to get their crosses set up by Wednesday. I checked in your bottles this morning. They are full of flies. So the Thursday lab, no, there aren't many adults yet, but there's lots of pupae, so you're doing good. So we should be able to get that done by Wednesday. And then we just have to sit back and let them mate and lay eggs all over the place in those bottles. Early next week, you will have to clear the bottles one more time. And then we wait patiently for the F1s. It'll be very exciting. So if you've noticed, I put up another video of what the experiment is. So review that and you'll know exactly what to expect in the next week. We hope. We'll see. We'll get whatever result we get. But I'm pretty confident this is going to work out. All right, so that's the lab this week. Oh, I said most of what you got to do is fly time stuff, but I will be in the lab at the regularly scheduled times. So if you just have questions, if you want me to take a look at your bottle and figure out if something's wrong or whatever, just grab me. I'll be there. Yes?
So basically, we don't have to come in unless depending on
right. Yes. I will be available, but it's all fly time this week, so you come in when you can. All right. The other thing is, there was an exam last week. Yeah, it's not quite graded yet because I just want you to know that I am generally very amenable if you need an extension. And we got a couple of students who asked for an extension. I gave them till Sunday night. So I'm going to sit down and grade those essay problems, short answer problems, this afternoon. So that'll all be done then. Okay. That means that if you didn't turn it in yet, immediately after this class, I'm posting the answer key on Canvas. And at that point, we close it off. No more submissions. I grade what's there, and that's it. Yes?
About how long do they take to grade?
How long will it take to grade? Most of the afternoon, I think, but by this evening I should have them done.
Oh.
Yeah, because Canvas does most of the automatic grading stuff, but I'll check their answers anyway. And it's 3 or 4 short answer questions. Piece of cake. I'll look through them this afternoon. Why, are you eager to find out about your grade? Yeah, okay. I know it's kind of frustrating because when the Canvas automatically grades the part that you can do, it gives you a score. That's not your full grade, okay? So if you think, oh man, there's another, I'm down to 50 points. There's another 30 or so points left waiting for me to grade. So don't panic yet. Panic this evening is okay, but no, we'll all be good. I glanced through them, they were looking pretty good. Okay, what we're doing today is something a little different. So instead of lecturing at you, remember last week I told you that you have now mastered Mendelian genetics? Who feels like they've really mastered it? I see heads shaking all over. Yeah, no, you've got the basic information in, but what you have to do to master it is practice. You got to do some problems. So we're going to go through some problems today, work through them step by step. I am also going to assign another batch of these kinds of problems for the homework that you'll get to do this weekend. Now we'll just keep this up until everyone's pretty comfortable with these problems. Okay, so let's look at some problems. Does this look scary? No, it shouldn't look scary. Okay, so this is This is a fairly simple problem. Where I say, okay, we've got a cross of an individual with the genotype big A, big B, little b, by one with the genotype big A, little b. And I tell you right out, the lowercase indicates a recessive allele. Then I ask you to figure out the phenotypes and frequencies of the offspring. Yeah, this will be a common sort of phrasing. Tell me what the phenotypes and the frequencies of those phenotypes are. Or sometimes I'll ask you about genotypes. Okay, but looking at this problem, let's start right away with breaking it down. As I told you last time, most of these Mendelian genetics problems can be crunched down to simpler problems. So this is a dihybrid cross, right? Because we got, here's one locus, here's another locus. We have an A locus and we have a B locus. And here we get the same thing, A and B. So as I told you, you can immediately start by just saying, okay, I'm going to keep this as simple as possible. I'm going to ignore B for a little while. I'm just going to solve the A part of the problem. We'll figure out the probabilities of the possible results there. Okay, does everybody see that so far? So let's just look at the A's. We pretend B doesn't exist, remember? And that is clearly this cross. Right? What will the results of that cross be? We're just asking for phenotypes. I told you we know Mendelian genetics, right? We memorized the monohybrid cross results. I have a list of names here. And if you don't answer, I will just start asking those names to answer. Let me just say, when I put you on the spot and ask a question, it is okay to say you don't know. I'll leave you alone if you just say, I don't know, and we'll try to explain it. Okay, so Amelia, are you here? No, Amelia. Is Raina here? All right, we're off to a great start. Okay. Let's see, Nigel. I don't see Nigel either. Okay, it's just the lower part of the alphabet. They're taking the day off. Let's see, Sam.
Stop.
Say that? Say again?
If you don't want to keep calling out names, I can.
Yes, I'll stop embarrassing yourself. Tell me.
Okay, so the result will be 25% big A.
Okay. So you're saying, okay, we get 25% this.
50% big A, little I.
Okay. Okay, and then you were about to tell me.
Yes, 25% little
Yes.
I, little I.
Very good. All right. However, notice the question. Phenotypes. So what is the distribution of the phenotypes?
3 to 1.
3 to 1. So these are all going to express the big A phenotype, and these are going to express the little a phenotype. So I'm just going to put this over here. 0.75 are going to be big A. 0.25 are going to be little a. Right? Yes?
Do we need to, if we express it in like a percentage form, is that going to be acceptable?
Oh, yes, you could say 75% and 25%. That works just fine. The catch is, we're going to do the B part next, then we're going to multiply them together.
Sure.
Yeah, so I'm just keeping it simple here. Okay, so the B part, what does the B part look like? The B part is big B, little b, by little b, little b. Somebody can answer that, right? Be brave. Go ahead.
That one would just be 50% big B, little b, and 50% two little b's.
Okay. I'm going to get this out of that cross. And 50%. Right? Right. Which, again, those are genotypes. What are the phenotypes we're going to see?
50.
Go ahead.
50.
Yeah. So we're going to see 0.5 big B and 0.5 little b. So then but now we've got two separate problems. We have to combine them, right? So the way I would do this is say, okay, we got these combinations. Little a, yeah, okay. So what is the percentage or frequency of big A, big B? You multiply those together, right?
Well, the phenotype would be 0.375.
Yes. So we got 0.375, or you could write 37.5%. That works. What's the frequency of big A, little b? Yeah, you just multiply those together, and that's going to be 37.5%. Okay, what's the frequency of little a, big B? Yeah, you just multiply those together. So that's going to equal 0.125. And then finally, little a, little b.
125.
Yeah, it's multiply that by that, and you also get 0.125. Now see, when you do this, when you break it down into individual pieces, and you can do that all the time in Mendelian genetics, it works out pretty easily. And then you just multiply them together to get the proportions of each of those. So I am providing the answer key. Oh, let me erase this so you can read it. And I will post this answer key once we're all done here. As you can see, I just start with just the A locus, then I do the B locus, and we get all these numbers. Those are the same numbers you guys came up with. You must have done it right. I also point out one other little thing here, that if you get this kind of problem, you can do a really simple cross-check. You add these up, and they better add up to 100%. If they added up to 110% or 80% or something, you did something wrong. Okay, that was easy, right? I think the key thing to get across to everyone is identify the loci. And this is a diploid organism. There should be 2 alleles present at each locus. So that's what we're seeing here. Okay. Also, I just did this, just in case people want to see it, is I hate thinking this way. This is the Punnett square version of that same problem. Now you could do this, and this will give you the same answer. Because, let's look for all the examples where it's big A, little b. There's 1, 2, 3, 4, 5. Is that it? 6, So we got 6/16 of those. And you could do the calculations that way. But you notice what I had to do there was go through and count each square, and it's tedious. I always find it much easier just to do the individual loci and then multiply them together, and you get a nice clean answer without likelihood of the kinds of error you would make of counting with these things. Okay. Here's another little story problem. So I've taken these straight out of the textbook. So this is kind of standard stuff. So I say here, albinism in humans is inherited as a simple recessive trait. For the following families, determine the genotypes of the parents and offspring. One thing I want to mention though, if you go to the book, it's got the same problems, but it phrases it a little differently. It says 2 normal parents have 5 children, 4 normal and 1 albino. Oh, seriously, come on. If you're working in genetics, human genetics, you got to be more careful in this. If you saw a family of 5 kids and one of them was an albino, would you say, oh, I see you got some normal kids, as well as a little weirdo over here, right? Yeah, so I've changed the phrasing a little bit. Instead of normal, I'm just saying wild type. I know we're not used to saying wild type about people, but yeah, wild type parents. And we get 4 wild type and 1 albino child out of this pairing. Okay, what's the genotypes of the parents? Yes?
They both have to have a recessive trait for albinism to have an albino child, right?
Right.
So they should be heterozygous.
They better be heterozygous. So yeah, so if we again use A for the symbol here. Another thing to note is often if I give you a problem like this, you get to pick the symbol, which makes it crazy and difficult for me to grade it, but okay. So we'll just say A. So we're saying the parents look like this. Big A is the dominant trait. They're all wild type. And then when you look at the children, how did you come to the conclusion that this has to be the genotype? What's the logic behind what you were thinking here?
Well, the result of double heterozygous cross would be 75% normal in appearance of phenotype, and then there would be the 1 albino.
Correct. So we'd expect out of this cross, the phenotypes you get would be 0.75 big A, 0.25 little a, standard 3-to-1 ratio. And you look at those numbers, that's not a 3-to-1, but we're dealing with very small numbers. People don't make 500 babies to give you good statistical tests. So we'd say, okay, and then of course the only way you could get an albino child out of this cross is if both parents contribute that albino allele. Okay. Next one. We've got a wild type male and an albino female. They have 6 children again. And 3 are wild type and 3 are albino. How would you interpret that?
The male would be heterozygous, but the female would have both recessive alleles.
Yes, because albinism is recessive, we know right away the female has to be this. Does that make sense when you get half albino and half wild type children? Yeah, that's what you'd expect, a 50-50 ratio from this cross. So that all works. Okay, the next one. We got a wild type female and an albino male. They have 6 children and all 3 are wild type. Somebody else? Or in this row. Somebody over here. Yes, go ahead.
Me?
Yeah.
Did I read the question
Yes, so I'm asking, okay, let's start with just the genotypes of the parents. What are the likely genotypes of the parents when we got a wild type female and an albino male and all of their children are wild type?
The wild type female would be big A, little a, and the albino male would be little a, little a. Maybe.
Okay.
Or, wait, what is the other one? But all 3 children are what?
But all the children, all 6.
The female would be double A, or double big A then.
Okay. Is this impossible?
No.
No, it's still possible, it's just unlikely. But yeah, the most likely answer is this. I would say you should probably also say this is a remote possibility. Tell that lady to go have 6 more children so we can properly assess it.
Oh, gosh.
No, we're not going to do that either. There's all kinds of restrictions on what you can do with people in genetics. That's one of them. Okay, that works. Yeah, there's the answers. Betsy, did I agree with the answers that you gave? Oh my gosh, you got it right. Oh, except we didn't go into detail about the genotype of the wild type children in part A. Maybe I should explain that for a minute. Okay, so we know the albino child is little a, little a. The 4 others, they're either big A, or big A, little a, right? Because we're doing this cross right here. If we ask for the proportions, my question didn't ask for, this one didn't ask for the proportions, but if I did, how did I get this number and that number? That's not 3 to 1. What's the logic behind that? Yes?
Out of the 3 and the 3 to 1, it's a 1 to 2 ratio.
Yes, but why? Well, we got the genotypes right here. So here's the thing, we got that, we got this. So in this kind of cross, what you will get, remember the ratio of genotypes, you would see this. Right? That's a monohybrid cross. We'd see those ratios. But what we've already said is, okay, we're just looking at the ones that are wild type. These. Oh, there's our wild type children. And the ratio within the wild type children is 1 big A to 2 big A little a's. Because we're excluding the albino children from it. All clear on that one? Okay. Here's another little problem. This one, like many genetics problems, it's a kind of logic problem. You know, that stuff you did on the ACTs or the SATs or whatever you took. There's those big sections where you're just logic problems. A lot of genetics is that way. So here I tell you, okay, pigeons may exhibit a checkered or a plain pattern, and we do a bunch of crosses. Darwin should have done these. I told you, he just collected anecdotes. But this is the kind of test you do. So what we're doing now is we're looking at the F1 progeny. So we're taking purebred, checkered, and crossing those, and we get that result. Purebred checkered by purebred plain, we get that result. Purebred plain by purebred plain, we get that result. So, first thing to do, make a hypothesis. How would you explain that distribution of numbers? Go ahead.
plane has to be tied to a recessive allele?
That makes sense. So whenever you cross a purebred plain with a purebred checkered, you always get nothing but checkered. And when you cross the purebred plain, so we got two cases where this happened. And when you cross plane by plane, that's the only time you see the plain pigeons come out of it. Okay, so that's our hypothesis. So our hypothesis is that checkered is dominant to plain. We'll see if that works. So we're not done with this problem yet, because the next thing we're going to do is we're going to do some crosses of these guys. We're going to take the F1s and we're going to get an F2 cross. There's a couple ways we can do that. So here I've just took that previous observation so you don't forget. It's kind of relevant how you interpret this next result. And we are saying, okay, checkered is dominant to plain. Here what you see is, okay, we're going to do the F1 crosses. And we're going to get some F2 progeny. In this case, what we're doing in D is we are crossing checkered, but this one by plain. That is that one. All right. Then we get the result over here, where we get 34 checkered and no plain. How do you interpret that? And by interpret that, I mean, what are the genotypes of this cross? Not the phenotypes, the genotypes. Go ahead.
The parents are both homozygous, and then the F2 is heterozygous. They're for different.
Okay.
Ones were
I see what you're, yeah, so this one from here, check it from here. This one, if I use P for the symbol here, this one is P, because it's an F1, which we obtain checkered by checkered, it's going to be P, big P, right? And we're crossing this to plane. We know what plane has to be if plane is recessive. Little p, little p. So we do that cross, and we're not surprised. We get all checkered, but the genotype of those individuals is going to be, right? Yeah, you all see how that works. Okay, the next problem. E, we got checkered, but in this case, we're looking at this one. Now we're crossing that to this one. How do you explain the results? 17 and 14. Go ahead.
The checkered would be heterozygous, and then the plain would be homozygous recessive.
Yeah, so this one, it looks like that, and this one has to look like that. And that's where you get the 50-50 ratio. All right. Easy, right? We're cruising through these. I think I got 21 of these problems here. I don't think we'll get through them this hour. But we won't run out. All right, so F, checkered B by checkered. Okay, we're going to cross this one by itself. Those two we're going to cross together. How do you explain that 28 and 9 result? Go ahead.
They're crossing two heterozygous parents, and so they're able to get both homozygous dominant and recessive, as well as heterozygous.
Yes. So we get our familiar 3-to-1 ratio. Okay, this all works. Finally, this checkered We're going to cross that one from A with that one. What are the genotypes there? What's the genotype for this pigeon? Say what?
Homozygous dominant.
Homozygous dominant, yes. So this one's got to be that. And B, that's this one. What's its genotype?
Heterozygous.
Yeah. So we cross those, and we're still going to get all dominant. We're still going to get all with a big P in there. Half of them, however, will be big P, little p, and half will be big P, big P. Okay. Ooh, look at there. We all agreed. But again, I went a little further and I said, okay, what's the frequency of these? 1/3 are big P, 2/3 are big P, little p, and this one cross. And you'll know now why, right? Same as the last one. Okay. Let's talk about fruit flies. Let's talk about a dihybrid cross. So we're out of that baby stuff. The monohybrid crosses, they're easy, right? So here's a dihybrid cross. So I've given you two different loci. One is gray body versus ebony body, so the E locus. And gray is dominant to ebony. And then the other one is long wings versus vestigial wings. The standard abbreviation for vestigial is little v, little g. So long wings are big V, G. We're going into new territory here. Because instead of single letters, we will discover there are so many different alleles out there. We got to go to 2 letters, or 3 letters, or 5 letters, whatever it takes to make a unique name, which will sometimes be a little bit confusing. I'll be talking about nomenclature later, and there are so many different mutants that a lot of times what they do is they go to superscripts. Here's VG number 1, here's VG number 2, etc. Again, we'll learn about that next week because we're going to play with some databases. It's as you might expect, because there's something like 15,000 different fly genes, 20,000 different human genes. You can't label them with a single letter anymore. That would only get us to 26. So we got a ways to go. Okay, so here I say, okay, we got gray body, dominant to ebony body, and long wings dominant to vestigial wings. And here what I do is I'm giving you the crosses by phenotype. Now here's another important thing to remember. If I give you a problem and I say, okay, here's the initial stock, assume they're purebred. If I don't say otherwise, assume they're homozygous at every locus. Okay, so we're going to assume that this is homozygous for gray, homozygous for long, this one's homozygous for ebony, this one is homozygous for vestigial. And we're going to get a cross, and we're going to get some F1s out of this. And then we're going to take the F1s and we're going to cross them to themselves to get some F2s. And we're going to ask, what do we get out of this kind of cross? Okay, so I'm going to go through each of these 3 one by one. So, up there at the top, I'm just reminding you, those are our little acronyms and our dominant and recessive relationships. We are going to ask you, what is the result in the F1 of this cross. Now, one of the first things you should do in any problem like this is write down what are the genotypes of the parents. So, what's the genotype of a gray long-winged fly?
Biggie, capital B, G, capital B, G.
Okay. Yeah, I know it's going to get a little confusing as the mutant names get longer, isn't it? But this is one locus. This is a second locus in a diploid organism. So at each locus we have two alleles to deal with. Okay, what about this one? What's the genotype of that individual? Go ahead.
All lowercase BE, BG, BG.
Yeah, and again, there's one locus, there's the other locus. Next step, we want to figure out, okay, this is the parental generation. All homozygous at all those, yes?
Can the gray one be heterozygous or no?
No, because this is the parental generation, which I've just told you is purebred.
Oh, okay.
Yeah, so just assume homozygosity in the initial stages of the cross.
Okay.
All right, so we're going to cross those. What will the genotype of the F1s be?
They're all heterozygous.
They're all heterozygous, so they will look like this and lowercase b. They look just like the uppercase ones there. So that's what our F1s would look like. Genotypically, what will their phenotype be? All of them dominant. What does that mean?
Gray long.
Gray and long. Yeah, so that's our phenotype. So that's what our F1s look like. Then I say we're supposed to carry these out into the F2s. So we're saying we're going to cross these with themselves. What will the F2s look like? And what will their frequencies be? You've answered a bunch. Does anybody else want to dive in first? Be brave. Again, remember what I told you earlier. Just break it down into the monohybrid crosses. Let's just look at EE. We're just going to look at the ebony versus gray bodies. So if we just look at that, what will you expect to see? Yes?
3 to 1.
3 to 1. So we're going to see 3 are going to be big E phenotypically, and 1 is going to be little e. So 1 is going to be gray, and 1 is going to be ebony. Or 3 is gray to 1 ebony. Or, because we're going to have to multiply these out, let me just do it this way. Right? Same thing. What about the vestigial ones? Vestigial versus long. What will we see in the phenotypes? Same thing? Yeah, we're going to see 0.75 are going to be long-winged and 0.25 are going to be vestigial winged. All right, but now we have to think about what about the proportions of the different combinations? So the combinations we have here is we could have this. So dark-winged or gray-bodied and long-winged. We could also have gray-bodied and short-winged. We could also have ebony body and long wings, or ebony body and vestigial wings. What's the proportion of each of those? For instance, to get that one, we multiply those two together, right? Which gives us what? 3/4 times 3/4 is a very familiar number in dihybrid crosses. Anyone remember? 3/4 times 3/4 is going to give you 9/16. Or you were calculating on your calculator the decimal value for that, right?
0.5625.
Yeah. Okay, I'll put that over here. Okay, same thing. 9/16, or 0.5625. What is the frequency of big E, little vg? That's 3/4 times 1/4. That's 3/16. Oh, that's point. You were about to calculate it, so go ahead and fire it.
0.187.
0.187. There we go. All right, then little e, big VG. So ebony body and long wings, this one. And multiply those together.
360.
Yeah, it's 3/16 again, which is 0.187. And then finally, these two. So 1/4 times 1/4 is 1/16. Notice the 9:3 to 3:1 ratios there. That's what you get with dihybrid crosses. A normal dihybrid cross, that is. That should just jump right out at you. So you didn't even need to do the math here. When I see this kind of problem, what I do is, the F2s are going to get a 9:3 to 3:1 ratio. Which one's which? That's what I'm showing here. So 9/16 will have the dominant allele at both loci. 3/16 will have the dominant allele at one locus, the recessive at another. 3/16 will have the recessive allele at one locus and the dominant at the other. And then 1/16 will be recessive at both loci. So that's how we get our 9:3 to 3:1 without doing any math. Once you know that 9:3 to 3:1 ratio, you can figure that out pretty quickly. Okay. You're all doing very well so far. So I can throw these kinds of questions on the next exam, right? No problem. All right, here's the other one. So I said we got gray vestigial crossed with ebony and long. Once again, take the problem and convert it to genotypes. Okay, we know these are homozygous at all loci. So what would you say were the genotypes here? Anyone who hasn't spoken up yet? I'm confident you can all do this.
Well, great is big E for the gray.
All right, so that's going to be big E. And vestigial is going to be what?
Small big EG.
Very good. Now we got an ebony and long fly.
Lowercase e,
And long will be big VG. Big VG. All right. What are the F1s going to look like?
All heterozygous?
All heterozygous, yes, that's going to produce. And then we're going to say, okay, let's cross those F1s to each other. Before I do that, let me step back. On the previous problem, we got exactly the same answer for the F1s, didn't we? Which means if you saw this on the exam, you could cheat and just say, oh, I'll just copy these down for this second. That would not be cheating. That's perfectly fair. So we will get exactly the same number here. This is pretty much the same problem except for the first cross. And there we go again. I just did a copy and paste myself. I think that's kosher. Go ahead and do that. Okay, we got one more in this set. Gray long by gray vestigial. Genotypes, please. What are the genotypes of those flies? They're purebred. So they're going to be homozygous. You can do it. Okay.
Is it big E for the gray,
Uh-huh.
and then big VG?
Okay, that's the first one.
Big E,
Uh-huh.
and then little.
Okay. So this is a 2-locus cross. But here's another simple thing you can do. When you see this kind of thing, you can just say, I can just ignore gray. All of the progeny of this cross are going to be gray. They're all going to be big E, big E. So I don't have to consider that. And that means this is actually just a standard monohybrid cross for purposes of calculation. So what will the F1s look like?
Heterozygous, and then gray.
So they'll be homozygous for gray, and then they'll be heterozygotes for wingling. And then I'm going to cross those by each other. Pretty sweet, right? Because we just know, we know for sure that all the progeny will look like this for E. What about for vestigial? What will we get?
3 to 1.
3 to 1, yes. So we're going, again, let's just go to phenotypes, because that's all the problem asks for. Yeah, the phenotypes we're going to see 0.75 are going to be this, and 0.25 will be this. So don't be afraid of these kinds of problems. The key things are, first of all, make it clear what genotypes you're looking at. We did that in each of these problems. Just from the description of the problem, you can figure out what these are. And then you break it down. You just say, okay, I'm going to deal with one locus at a time. So in this case, the body color locus was easy. And we just have to consider the wing length locus. And so we get that nice 3-to-1 ratio here. Okay, there's the results if I unclutter my screen a little bit. Okay, let's do another one. Oh boy, chi-square. I think we got time. Did I hear a groan? Oh, yeah. Chi-square, again, is really easy, but it's kind of tedious, right? You got to go through all these steps. So what we've got here is two of Mendel's monohybrid crosses. So we're going to cross full pods by constricted pods. Now we get 882 by 299. Okay, we ask you for a couple things here. Tell me your hypothesis to explain that result. Then calculate a chi-square value. Why do you need to get a hypothesis first? Because that's how you figure out what the expected numbers are supposed to be. So we got our observed numbers. We need to have a hypothesis so we can figure out, okay, what did we expect in this cross? And then we grind through the old chi-square analysis. So let's do that. See how nice I am? I just put it in a grid for you. I won't always do that on an exam. But here I'm putting it right there in a grid. They say, okay, we've got 822 or 882 full pods, 299 constricted pods. What is our hypothesis? This is basically which one is dominant, which one is recessive. Full pods are dominant. So what ratio should we see? 3-to-1. So we need to see a 3-to-1 ratio. So we got to figure this one out. How do you figure out what the expected was? You look like you're itching to raise your hand.
Maybe add up the total number of pods and then
Okay, add those up.
find the 3-to-1 ratio.
Well, yes, you're going to get a number X here. Somebody figured that out for me.
For the expected, for the
Oh,
constricted.
okay.
We're not doing, or we just doing.
No, go ahead. Go ahead and tell me the whole thing.
Okay. Wait, do you want me to tell you the total or the?
Tell me all of it.
Oh, so for the expected, for the constricted, it'd be 295.25.
295. And how many of the full pods did we expect?
885.75.
Oh, wait, but
The other one has a point.
295.25. Yes, okay, that's better. All right.
Oh, sorry, 885.
Oh,
Not just 882.
yes, okay. Transcription error. Okay, so there's what we expect. And again, easy to calculate. You just had to add these up. You get a number, the total that you multiply by 0.75 and 0.25, and presto, you get your expected numbers. Then we're going to grind through those other steps. Take the difference, square it, divide it by the expected. I'll let somebody else tell me what that answer is.
For full, at the very end, it would be 0.0158.
0.0158.
So you have, because you have to. And then this would be the ratio.
And then you times it times by 3.
Okay, what about constricted? Did anybody figure that one out? 0.0476. All right. More significant digits than we need, but that's not going to hurt. And then to get the chi-square value, we just add those up, right? That sound right? Yeah, not that much. Just eyeballing it. I'd even accept 0.6. We're just looking at the ballpark here. Okay, so our chi-square value is 0.6. How many degrees of freedom do we have in this problem? 1. Yes, just 1. So we just need to look right here. Where does 0.6 fall in that table?
Between 0.05 and 0.01. Or wait, no.
No, it's a 0.64. It's a tiny number. It's going to be somewhere over here, right? So P is going to be pretty big. Well, we can just say, okay, P is greater than 0.05, or P is greater than 0.2, since that's all I gave you here. So what does that mean? Does that mean these numbers fit with our hypothesis? Yes. So we would say any variation that we see here is just due to chance. It's not some kind of systematic difference from our hypothesis. All right, did I agree with you? Yes, I agreed with you. All right, oh wait, how did I get 0.064? Oh, we got a decimal point difference. Oh, I will double check my numbers later. But okay, so we do this. We get a tiny chi-square number, which means we got a big p value, which means that any deviation is the product of simply chance. Okay, let's get rid of these. Because we had another one, didn't we? Yeah, it's a little bit grindy, I know. But you guys tell me what the answer is. I'll just sit back here, be comfortable. First of all, what's our hypothesis?
Violet follows are dominant.
Yes, okay. So we expect a 3-to-1 ratio. Let me know when you got a chi-square. Yes?
I got 0.39.
0.39. Let's see, what did I say? Oh, right on the money. That's what I said too. And if I said it, you must be right. Yeah, so chi-square is about 0.39. And once again, we got 1 degree of freedom. We go down to our little table here. That means that chi-square is somewhere off to the left here. So that means that P has to be greater than 0.2, or all we care about is greater than 0.05. So once again, this fits pretty well. It accommodates chance pretty easily. Now, I mentioned before that Mendel had some critics who said, no, this is too close. Look how close those numbers are. Yeah, these are really pretty much spot on as far as I'm concerned. Yeah, so there's a difference of about 8 from the expected almost 700 here. Do you think he cheated? You're confident. Why not?
Because it's a good chance, and you have like the numbers are quite similar.
Yeah, so that tells us that his observed numbers are really close to 3 to 1. There are other statistical tests we could do, and we could say how likely it is that we get this close, which you don't need to know for this class. Isn't that nice? If you know chi-square, you're good. So this is surprisingly close, and it made people very suspicious. And as I mentioned before, I personally don't think he exactly cheated. He probably cherry-picked his data a little bit. But then it's been tested and tested and tested and tested for 150 years since, and it seems to be holding up pretty well. So I think now we've got a better estimate of this. Okay, here's a question for you before I let you go. Shall we do some more problems on Thursday, or are you ready to move on? A shrug is not an answer. Do you feel pretty confident? Okay, so maybe what I'll do is we're going to talk about some other stuff on Wednesday. Yeah, I'll lecture at you again. And I'll give you a problem set to do over the weekend. And if you screw it up, I will punish you by giving you more problems to do in class. We'll make sure everybody has mastered this stuff. Okay, so we'll see you on Wednesday then. We have to talk about pedigrees.